Good day, everyone, and welcome to our seminar. The seminar is on writing a thesis in sciences. Several students across universities are required to write a thesis before they graduate from their program in partial fulfillment of the requirements for the award of their degrees in sciences. And so, the seminar is on highlighting the important things that made up a thesis in sciences, irrespective of the departments or the faculties in sciences. There are at least major things that the thesis in sciences have in common, and this is what we are going to discuss in this seminar. We have components of a thesis in sciences. Most theses are organized in 5 chapters. Most theses in sciences are organized in 5 chapters. And so the components that made up these 5 chapters of a thesis are: number 1 is the introduction, number 2 is the literature review, number 3 is the materials and methods, number 4 results, number 5 discussions. You then have references, appendix, and an abstract. So we are going to look into these components of a thesis in sciences. We are going to start with the introduction. The introduction is usually the first chapter of a thesis. Under the introduction, there are things that made up that chapter or the introduction. First, you need to have a brief overview of the topic that you are writing a thesis on. So you have to make a brief introduction on the topic that you have chosen to write your thesis on. And after a brief overview, there is what is called statement of the problem. Of course, every topic in sciences seeks to address a problem that is existing in the society. And so, if a topic that you have chosen to write in your thesis does not seek to address any problem, then that topic needs a review. That topic needs to be revisited. So there must be a statement on the problem which your topic seeks to address, or a gap in which your topic seeks to close. And then there is a justification for the study. After stating the problem, there is need to justify why you need to spend resources on that topic. So there is need to make it clear that the investment into that topic, the finances, the funding, the time, the effort that you will put in writing that thesis will not be a waste. That is what is called justification for the study. Then after the justification, you have to state the aim of the study. Every study has an aim. That aim serves as a direction towards which the thesis is going. That has to be defined at the onset of the thesis. If not, the thesis may be going left or right without having a common purpose. Usually, in most places, the topic itself is usually the aim of the study. That particular topic of the thesis is usually the aim of the study in most places. And then there is specific objectives. Specific objectives is usually a subunit or a subsection of the aim of the study. When you have an aim, you have to break down the aim into 2 or 3 smaller units. And those smaller units will help you to achieve the major aim when you achieve them individually. When you achieve the specific objectives individually, you can now put them together to become the aim of the study, which you seek to achieve. And so the specific objective may be 2, it may be 3, or it may be more, depending on the nature of the topic. In some places, there is also what is called research questions that is raised at the onset of the study, which the study seeks to address. And so it's expected by the time you finish your entire studies, the practicals that are involved, that all the findings you make from the field, from the laboratory, will give answers to the questions you have raised at the onset in the introduction. So this is a general overview of what the introduction looks like in a thesis in science. Now let's talk about the literature review section. This usually forms the chapter 2 of the study, and as the term implies, literature review involves reviewing published academic journal articles and textbooks that are relevant to the topic. You review them, and then especially how it relates to the topic that you are working on. Most times, most recent journal articles are preferred when seeking articles to review for your study. Most recent articles are articles that are published within the last 5 years as at the time you want to embark on your study. They are mostly preferred, but that does not mean that someone who is writing a thesis cannot review articles published 10 years ago or 20 years ago if they are relevant and important to that topic. But most often, it is required that the majority of the papers you review will be those who are published in recent times, maybe not more than 5 years. That is what the literature review is all about. And then the next component of a thesis in sciences is materials and method. Under the materials and methods, you have to describe several units. I've listed them on the slides. The first thing is description of the study area. The second is the study design. The third is determination of your sample size. If you are using samples from humans. And then the ethical clearance, advocacy visit and sensitization, instruments and validation, then sample collections and experimentation, and then data management and analysis. Let's take them one after another. In the first one, description of the study area, the researcher have to describe the area, whether it's a laboratory or a field where the study will be conducted. Some of the things to describe include, if it is in the field, the population of the area, the number of people living in that area. And the population of the people is also relevant when you will determine your sample size to know exactly how much persons will be able to participate in the study from that population to represent the entire population, because there's no way you can work with the entire community. No. And so the description of the study area can be describing the population, describing the features of the area. The features include the environment. You can also describe the vegetation of the area. You can describe their climate, the temperature, the relative humidity, and other things that are in the study area which are relevant to the topic that you are working on. And so after the description of the study area, the next thing is to determine the study design that you are going to use in the study and state them boldly. The study design broadly are in two major categories. And the first one is descriptive and analytical. And so under descriptive, there are smaller units, and under analytical also. But this is the broad category of the study design. And so the study design must be known. The study design must be known. And then determination of sample size is done using a formula or several formulas. And most times, one formula may be enough to determine the sample size if most times they are human samples. You think, for example, you want to study in a community of 100,000 persons. Let's assume you're working on malaria parasites and you need to collect blood samples. There is no way you can collect blood samples from 100,000 persons in a community. But using a sample size formula, several of them, choosing the one that is right and suitable for you, you now use it to calculate your sample size based on the actual population of the study population, and then the sample size you have after calculation is now what you use to work, which is your sample size. These are the ones you collect sample from that you use for the study. But it's good to also state that sample size determination is usually so much important when working with human samples. It may not be the same when working with other samples that are not human. Then also when you are working with human or animal samples, you must obtain ethical clearance from ethical committee. We have ethical committees for human samples in several hospitals, and we have animal research ethics committee also in several places, mostly in universities. So if you're working with human sample and you're working with animal samples, you must obtain ethical clearance from a committee in charge of it. This committee is in charge of reviewing your proposal and then approving if your studies will not be harmful to humans or if they will not be harmful to animals. And it's until you obtain your ethical clearance letter from such committees that is only when you are advised to start the study. Also, there is what is called advocacy visits during practicals. Advocacy visits and sensitization. This is very necessary when you are collecting your samples from a community. It is not proper to move into a community without notifying anyone. You have to go for advocacy visit ahead of time before the study, before sample collection, and visit the opinion leaders in the community and explain yourself, explain your study, explain how your study is going to benefit the community, and then sensitize them and come to a mutual agreement that you can carry out your sample collection or whatever as relates to your study in their community so that you'll be able to progress in your thesis writing. There's also instruments and validation. Every study in sciences involves instruments in the laboratory and instruments in the field. For example, is the microscope and a whole lot of other instruments. Even the questionnaire that you will use if it involves a questionnaire study is also an instrument that needs to be validated by a professional. There's a danger in not validating instruments. Take, for example, you are using a stopwatch which you did not validate. The stopwatch may be malfunctioning or the microscope may be malfunctioning and no one knows. And then you use it like that and obtain results that does not represent the true situation in the study area. And so when the instrument introduce error during the analysis, the error will definitely manifest in the results that you obtain. And of course, that study will no longer represent the true outcome of what it should have been. So there is need to validate the instruments, both in the laboratory and in the field, and confirm that they are working effectively before you deploy them for the studies. Then there is also sample collection, processes, and experimentation. This is where the one who is embarking the study to write the thesis will have to collect samples, whether blood sample, whether urine sample, whether stool sample, whether all forms of sample from humans or from animals or from the field, wherever the samples are to be collected. There is also need to describe how the samples are collected in this section of your thesis. There is also, it is also very important to explain the experimentation processes used to obtain, after collection of the samples, to continue in the study. So the experiments should be described in a clear term that is not ambiguous to understand. And everything you do with the sample and the experiment will yield data. So you have to collect the data using softwares such as Excel to impute the data, and then use other softwares that are for analysis to analyze the data. Data management is very necessary, and data analysis is also necessary. If the data you've collected cannot be interpreted statistically, then the such data may not be useful in sciences. So there is need to manage data properly by entering them in appropriate softwares or sheets such as Excel, and then analyze them with appropriate softwares. We'll now talk about the results. When you have obtained your data and analyzed them, it gives rise to results from the study. And so in the results, in this section, in chapter 4, you have to present the results with clarity so that it will be very, very clear and understandable to anyone reading them, even people outside your own field. So you can present your results using tests. You can present them using tables. You can present them using charts such as bar charts, pie charts, and others. You can also present your results using graphs such as line graph. But one thing is important. It is wrong to duplicate results. What do I mean? To present the same result in two different ways. After using tables to present them, you now use charts to also present the same result, or use graph to present the same result. No. If you use tables to present a result, there is no need to also plot a chart for the same result. That is duplication, or to plot graph. So you choose either you're presenting the results with tables, or you're presenting them with charts, or you're presenting them with graphs. Now, we'll talk about the discussion section. In the discussion section, you have to discuss the results you have presented in the previous chapter, that is chapter 4, comparing them with relevant published academic journal articles, which you have earlier reviewed. Compare different aspects of your results and specific objectives that you have achieved using the findings from published work of other people who work in the same thing that you are currently working on. So you have to discuss your results with citations, citing other people's work in tests in your work. And that's way, after comparing them, you will now be able to make your own opinions as regards to your current findings. And in chapter 5, when you are done discussing your results, you also have the conclusion section where you make conclusion from all the findings. And then you go and make recommendations from your findings. What are you now recommending? Are you recommending further studies? Do you have other practical recommendations that will help solve the problems you identified earlier in chapter 1, which your result has already met some findings? You now make recommendations that will help either non-governmental organizations or governments or even individuals who want to intervene in your study area or in the area of your thesis to know exactly what they are going to do. And that is the last chapter for the thesis. And after that, you now organize the reference section. Every paper you reviewed in your literature review section and every paper you used in your discussion in chapter 5 or papers you used their methods in chapter 3 to also carry out your own experimentation. The full bio of all the in-text citation from chapter 1 to chapter 5 will now be listed in the reference section in full, including where they are published, the journal names where they are published, the volume number, the page numbers, and even the digital object identifier number also need to be there if it is available. Then you have appendix section after the references where you put all the approval documents you collected for your study, including the ethical clearance letter where applicable, including the identification letter where applicable, and then all the raw data that was generated during the analysis, the data analysis, even the pictures you took in the lab and in the study area will now come in the appendix section. After the appendix section, you now organize an abstract. What is an abstract? Is usually 300 to 500 words, as the case may be, which is a summary of everything that you did, everything you wrote in your thesis, from chapter 1, which is introduction, to literature review, to chapter 3, methodology, and to results in chapter 4, and your discussion in chapter 5. A summary of everything between 300 to 500 words will form the abstracts of your work. You summarize them. Mostly in journal articles or in thesis, the abstracts usually appear first before the introduction. But in the actual sense, or in the real sense, the abstract is written last after you must have concluded everything about your thesis, but is usually placed first. And so these are the vital informations on how to write a thesis in sciences in partial fulfillment for the award of various degrees in sciences across universities.